Hi, my name's Rachel, and today we're going to talk about the bacteriophage.
So normally when faced with a bacterial infection, what a physician is going to do is they're going to prescribe an antibiotic, but with the emergence of antibiotic-resistant bacteria and superbugs, for example, some common ones are VRE and MRSA, this kind of treatment is just really becoming less effective, which is unfortunate, but a promising and very, very exciting alternative would be the use of the bacteriophage in human therapeutics. The word bacteriophage means bacteria eater. It comes from the Greek word phagein, which means to devour. And they were actually discovered in the early 1900s. They were discovered by two different scientists, but they never became widely used due to the subsequent discovery of antibiotics. There were some studies with them conducted in Eastern Europe and the Soviet Union, however, but they occurred around the time of the Cold War, and a lot of their findings were not translated into English or even available in the Western scientific community until several decades later. So basically, a bacteriophage is a virus, and what it will do is it will actually infect bacteria in order to replicate itself. And there's so many different types, they're all very diverse, but they all have certain structural characteristics, one of which is the head. So the head consists of the genetic material, which will be either DNA or RNA, depending on the phage, surrounded by a protective protein coating. And then the next structure is the tail sheath. The tail sheath is basically a tunnel which allows the DNA or the RNA to pass from the head into the bacterial cell upon infection. More complex bacteriophages will have structures like a base plate or tail fibers, and these will help it attach to its receptor on the bacterial cell wall. Another way to think of a bacteriophage would be as a predator for bacteria. Wherever one might find bacteria, there's a high likelihood that you would be able to find a bacteriophage there as well. In addition, as a virus, a bacteriophage can mutate. This is so incredibly important. Let's say that a particular bacterium becomes resistant to a particular bacteriophage. This phage can change, it can mutate, and it can evolve so that it will be able to infect that bacterium again. This will prevent the kind of resistance issues that we are facing today. A bacteriophage can undergo one of two different life cycles. The first is the lytic cycle. This begins with attachment of the phage to a receptor on the bacterial cell wall. This requires a precise molecular interaction, and for this reason, phages are incredibly specific. Then, the bacteriophage injects its nucleic acid into the bacterium, and this is used to hijack the cell's biosynthetic machinery and to produce new viral parts, such as proteins and nucleic acids. These are then assembled, and eventually the viral lysozyme destroys both the bacterium and the bacterial cell wall, allowing these new viruses to burst free and infect new hosts. In the lysogenic cycle, the phage DNA is integrated into the bacterial genome. This creates what's known as a prophage, which if activated in the future will initiate production of new phages. Thanks for watching.